So before we kick off, we'll be asking each of the panelists to give an introduction to themselves. So David, please take a few minutes to tell us about your background.
Yeah, they gave us 5 minutes each to introduce ourselves, so you'll have to bear with me for a moment. My name's David Van Hout, and to start off, I thought, since we're in Boston, it's good to be in Boston, actually, and we're in Boston, and Oxford Nanopore is a UK-based company. I thought I'd give a little family history and joke. So my last name is of Dutch origin, Van Hout, right? But my maternal side is actually from England. Came here in 1609 to Jamestown, eventually settled in Boston. There's actually a few streets by Faneuil Hall still that bears our name. But they had to go to Nova Scotia because they were loyalists during the American Revolution, and then they couldn't return back to Boston, so we settled in Saratoga, which trails right into where I am stationed. So I'm in our manufacturing site for Regeneron Pharmaceuticals. It's outside Albany, New York, which is very close to Saratoga, if you know Northeast geography. As was said, I've been there almost 10 years, kind of clawing our way to validating and implementing NGS-based assays. To really, our main focus was adventitious agent detection-based assays using NGS. If you were at some of the previous sessions earlier, you've heard that they've updated the regulations. It's really kind of a watershed moment, let's say. And there's a lot of submissions now, so we hope to be one of those submissions next year. So yeah, we are mainly in the business of hunting for viruses, like I had mentioned, trying to automate all steps of that process, which we could probably talk about. But the other half of what we started getting involved with, like a lot of biotech companies, right? Regeneron's looking more into cell and gene therapies. So my group, based on our NGS experience and building out, bioinformatic software engineers, we've been diving more into, gene therapy. Specifically right now, mainly AAV-based therapies. So we have a lot of experience there, and I'm sure we'll talk about that. I think I'll stop there.
Fantastic. Thanks so much, David.
Yeah.
I was boasting to you about my family tree going back.
Yeah, I didn't tell you.
And you didn't even tell me, so there's going to be more questions about that over coffee. Sebastian, now over to you.
Okay, thank you. Thank you, David, for setting the stage. I have no clue what my family did in 1609. I can just tell you that I'm German and American. I have an American mother, but I'm born and bred in Germany, so my accent gives it away, and that I'm maybe a little bit more German. And so good afternoon, everyone. Before we start, I need to outfit myself with a little gimmick that I brought, and I hope that everyone, at least in the first rows, can see that I'm wearing a hat that says PDA, Parenteral Drug Association. So, as Kaz said, my name is Sebastian Teitz, and you might be wondering, why is Sebastian wearing a PDA hat right now? And that's because Veronica here, hi Veronica. So Veronica asked me to figuratively wear a PDA hat to report out the learnings around NGS from the PDA virus conference, which is, I think, the most important conference on the virus safety of biopharmaceuticals in the annual event calendar. So I'm serving on the scientific planning committee there, and so I wouldn't be me if I wasn't taking the hat thing to a literal level. My mate Josh at the PDA outfitted me with that one. I want to give out a little disclaimer. I'm an SME in virus safety of biopharmaceuticals, yes, but that is more on the DSP and the downstream clearance part, and for the deeper technical sequencing questions like how to curate your databases, etc., I would like to refer to David and Gaston, and I'm basically here to reflect on the learnings and the discussions that we had at the PDA virus conference. Now, what is the PDA? The PDA is a volunteer and individual membership-based organization, and that means that everyone can partake in the industry stakeholder activities regardless of your affiliation. And that is quite a big difference to other organizations, I would say. It's a non-for-profit organization, and where you can engage in interest groups establishing best practices on focus topics. And that is, one that is worth mentioning here in this context is certainly the Adventitious Virus Detection and Testing Working Group that promotes adventitious agent testing by net methods. That's what this working group is doing in a nutshell. So there are also local chapters for regional interactions and, of course, dedicated conferences and workshops where the state of the art of a subject is discussed in depth. And one little note, PDA is putting out more technical documents and scientific publications than every other for-profit organization, as much as I know. And those are peer-reviewed technical documents which serve as a very good guidance to the industry. So the stakeholders that are active here are people from authorities, manufacturers, suppliers, and CROs. And one unique selling point is that engagement of the regulators is basically guaranteed and I think higher than anywhere else. So here you really have the chance to directly interact with those persons and ask them your questions. So last thing, the PDA's slogan is connecting people, science, and regulation, and that is actually true in this order. So please do feel invited to partake. So now I'm here to report out the PDA Virus Conference learnings, and this year's edition was, of course, focused on the revision of the ICH Q5A guideline, the overarching guideline for virus safety of biopharmaceuticals. And a lot of focus was put on the aspect of how it enables the implementation of NGS and PCR-based test methods for the sake of adventitious agents detection. This was a central topic of discussion at the conference. So the ICH is now adopted since this year, and it explicitly promotes the broader application of NGS and PCR-based technologies for detecting potential virus contaminations. Now it will please many industry stakeholders to learn that the revision gives a really good guidance on many aspects of virus safety, and it also facilitates implementation of NGS in numerous ways. For instance, we learned that the classical adventitious agent test, they may be readily replaced by molecular methods. And the good news is that these alternate tests, they do not need to go through a head-to-head comparison, for example, with animal tests, and thereby reducing ethical and sustainability concerns by sparing numerous animal lives. So consequently, we are not only reducing the consumption of animals during a medicinal product's life cycle, we can actually start right at the beginning, right? So there's actually quite a lot of simplification, which is much needed in an ever more complex world. And there's some beauty to this revision. So I'm looking forward. Let's discuss here about how we can implement that. And thank you. I'm looking forward to an exciting exchange here.
Thank you so much, Sebastian. Loving the hat. And now over to you, Gaston, online.
Hi there. Hi. Nice to see you all virtually and from the back, because the camera is actually at your back. So unfortunately, I indeed couldn't attend today because we're so very busy at establishing assays using ONT for our mRNA therapeutics business unit. Without knowing David and Sebastian, I do see a bit of connection because I'm working at Lonza, Netherlands. I'm also Dutch, so there's a bit of connection to David. But for the last 10 years, I've been living in Munich. So also, again, a bit of connection to Sebastian, where I've had a great time in Germany as well. And so my background is mainly, I'm a medicinal chemist, so my roots are in organic chemistry. And the last 10 years I've been basically working at Roche Diagnostics for therapeutic drug monitoring. And I'm fairly new to the oligonucleotide field. And since I took up on this position at Lonza, we've been facing many different challenges as the analytical load for these therapeutics is pretty heavy. And pretty heavy, that means basically we're looking at 30, 35, sometimes up to 40 different assays that we have to establish for these kinds of therapeutics, not only because of the oligonucleotides, but also the way they are being packaged, I'd like to say, in lipid nanoparticles. Yeah, so I'm also very much looking forward to all your questions and to see how we can work together.
Fantastic. Thanks, Gaston. That's fantastic to hear from all of you and how you're working with the technology. I really want to make this an interactive session this afternoon. We've got a real opportunity for a rich dialogue, so please do submit your questions via the QR code, or we'll take them live in the room too. Before we kick off with any questions in the room, I'll just take the opportunity to ask a couple of questions to each of the panelists to kick us off. So, Sebastian, we heard in the previous session about a GMP validating nanopore-based AVA test. Can you give the audience a feel for how the regulatory space has really changed recently to facilitate the introduction of the use of sequencing-based methods?
Yes, of course. Thank you for the question. If I may, I would like to quote a character from one of my favorite TV shows, Futurama, the crazy scientist that always says, "Good news, everyone." But in contrast to the TV show, this time it's really actually good news. So what we have seen is that many companies were a little bit reluctant to implement these alternate methods to in vivo, in vitro testing. And according to one WHO survey quite recently, a lot of companies were actually waiting whether that ICH Q5A revision would really state that...
We were waiting.
Yeah, okay, yeah, good to know. So they were actually waiting. Will the revision really state the replacement of these classical methods by NGS and PCR-based testing methods? And now that we know it happened and the revised guideline is in force, we are seeing companies taking up these options quite speedily. And if you want, I can give you a couple of numbers later on how much that is.
Right, fantastic, thank you. And David, you mentioned that you're in the process of GMP validating a nanopore-based AAV test. Can you give the audience a feel for how nanopore is performing for those CQA assessments that you require for the product filing?
Yeah, the short answer is that it performs very well. I forgot to mention in my intro, we do have most sequencing platforms, so we do some short read, we do nanopore, and we do some analysis on PacBio as well. But I think to answer your question, yeah, it performs really well. We're getting quality that we need. I can give you a little case study. It happened this week, actually, Monday. Currently, for some of our more advanced clinical products, since we're not validated yet, we use contract labs to do some of the testing, and they're using a certain short read technology, and it actually couldn't. There was enough depth, but the last two base pairs of each of the therapeutic did not meet their own quality thresholds. And on our spec, we need 100% homology, and it didn't come back that way. So, however, we do get consistent quality using nanopore. We've never had that issue before. So I think that's and this is Monday I'm talking about. So a very recent case study to show that it is performing very well. In addition to that, with just the new technology and sequencing in general, we can, I term my assay as, one assay to rule them all. So we can really determine all these. It's my Lord of the Rings joke. Everybody told you I was going to tell you that. Yeah, you can really get all these different attributes, tests, metrics in one test session. And it was kind of mentioned earlier, and I've heard it in a few talks. That's kind of the idea to incorporate multiple tests into one test session, reduce cost, reduce turnaround time, and obviously you're getting a lot more information.
And what do you think the sort of significance of your findings are for the rest of the industry to sort of implement this technology? Yeah, so good question. I actually think it's pushing the industry and the regulators actually to rethink of what their requirements are. So just piggybacking on what I just mentioned,
historically, the requirement for AAV therapeutics really was just to identify the gene of interest, right? But now with these new technologies, again, we're getting so much more information about the ITRs, and now the requirements are changing. So they want to know about truncations. They want to know ITR to ITR. Before, the ITRs, no one really cared. Just show us the gene of interest, and that's really changing. So I think technologies like Oxford are really helping drive changes from that end.
That's fantastic to hear. Gaston, over to you. A little bit of a different theme for mRNA QC, whereby sequencing really was essential for critical quality attribute, it's particular to identity. So how do you see the analytical measurement of mRNA being streamlined using nanopore sequencing?
Mm-hmm. Yeah, so I think still state of the art is still Sanger sequencing. The old-fashioned way to do it. But of course, to have a sequence from RNA, you first need to reverse transcriptase it into cDNA before you can do the sequencing, right? So if you see something then peculiar in your sequence, well, then the question arises: is this now coming from the RT, so the sample prep basically, or is it something real? And then you basically need to redo it or go back into your data, look into that. So the direct way of looking at a sequence or sequencing is, of course, supposed to be more accurate, right? And also when we compare the two different techniques, and you mentioned streamlining, well, of course, if you don't have to go via reverse transcriptase, then you're also much quicker and cheaper. So in a way that really helps a lot to streamline those things. And also worth to mention, I think, and I think that's the great hallmark here, is that we are together with ONT working on a workflow that not only captures sequence or identity, but also three of the other very important critical quality attributes, namely the poly-A tail length and 5' capping, as well as integrity testing. And so if we can capture all that in what we call a multi-attribute method, then of course, well, we significantly lower the costs and the time that is needed to get to all those different results that are also not only our customers are asking for as a process development routine analytics, but also the authorities are requesting, of course. So yeah, in terms of streamlining, I think we really look into dozens of hours of less work, and therefore also the release times of these kinds of therapeutics is much less. And I think that's really important to keep track of time, especially with these kinds of products that are inherently already less stable than compared to DNA, for example.
And what, maybe this is a question for all three of you, what internal education have you kind of gone through at your organizations to really successfully onboard nanopore sequencing? Maybe to you first, Gaston.
Mm-hmm. So what kind of education you mean? Or,
yeah, in terms of like training or getting people aware of nanopore and how to embed it really into your organization.
To be honest, we have people in-house that have many years of experience with various sequencing techniques, also Oxford nanopore sequencing. But then I think we do also train people anew that have zero affiliation or zero experience with sequencing, and they also seem to grasp everything in 1-2 days of training. And because it's what we're aiming for is a very plug and play work quality control. So the workflow is mostly we set something up in analytical development, and then the further validation, routine use happens in quality control, where those people are not always experts. Because they simply are also in routine use, and they also need to be able to run an assay in a couple of hours without in-depth knowledge.
Yeah, fantastic. And what about for you in terms of internal education, both for maybe challenges in getting people on board with moving, pivoting to different technologies?
Yeah, we've had a challenge just kind of convincing our own institution, as you know, Sebastian was mentioning. We were waiting for guidance. You know, it kind of was why are we investing this right now if it's not even going to be accepted? So it was kind of a push and pull back and forth. But we just did it. When I started at Regeneron, there was already a MinION on my desk in 2015. You know, so we've been playing with it for a while. It's just a fun tool, actually, in terms of some of the other platforms. So we've been early adopters, and you know we now have GridIONs and we're getting the Q line. It's just been really easy, or not easy. We can see the potential of it, and we can see that we can automate it, the sample prep, potentially automate even the loading of the flow cells, yeah, so we just chugged ahead.
Yeah. And maybe to you, Sebastian, the sort of education for the
Yes.
regulators, how has that journey been? And can you kind of maybe comment a little bit on that?
Good. Yeah, right. I mean, yeah, that question pertains to me in a different way, not because I never worked in a organization that needed NGS in any way for their daily business. But what I witnessed is like the education of the whole industry on this topic and also of the regulators. And actually, the first time that I remember that I really heard the term NGS, that was 10 years ago, just across the river at the MIT at one of the CAACB meetings. And back then, the question was when do we think NGS is going to be used in routine QC testing? And well, some were quite optimistic. Some said 5 years from now. That we didn't we missed that by quite a long shot. But yes, so the industry is getting more experience with that. The regulators get more experience with that. I think they know how to phrase their questions and what they want actually a little bit better, like these validation package, doing spiked runs with your material and stuff like this, and the mutual understanding is becoming better. And actually, when we go to products that are really new from the technology point of view, like cell therapies, for example, they don't come from well-characterized cells like CHO cells, right? So what we need to do to bring cell therapies to the market is we need to characterize them from scratch, de novo, and that cries for NGS methods, including the ONT technology.
Fantastic, thank you. So I'm being quite selfish and taking up the time with questions from myself, so I want to open it up to the floor. Does anybody before I take the ones from Slido, does anyone in the room have a question? There must be some burning questions. Everyone's being a bit shy. All right, I'm going to let you have a think about it. I'm going to come back to you. You're not getting off that lightly. But I'm going to take one from Slido because it's been upvoted. This is one just for you, Gaston. To what extent has Lonza implemented ONT sequencing into their RNA analytics, both RUO and GMP? And do you see this changing in the future?
Yeah, so together with ONT, so there's a collaboration with ONT where we work together on implementing the technology for the critical quality attributes that I just mentioned. And so foreseeable, we will be able to, in a staggered approach, validate those methods by 1-2 years. So I think it's fair to say that in 2 years we will be able to offer these methods as a platform multi-attribute method to our customers in a GMP fashion. So meaning validated by QC, and platform in our terminology basically means that these assays have been validated for 3 different RNA products, And what we then offer to our customers is basically an onboarding onto the platform. And that entails that if their product falls within the same design as for which we did the validation, then that kind of assay can be offered without any validation anew, which of course again also lowers the costs for our customers.
Fantastic. Thanks, Gaston. Anybody else in the room? Oh, we've got one at the back.
Yeah, this is a great session. The world of, I guess, GMP regulated testing is pretty new to me. So apologies if it's a very naive question. So wondering in terms of, I guess, traditional testing like sterility or mycoplasma testing, which might take like a month. When it comes to acceptance by the regulatory agencies, there is a sense that traditional ways are the way to go. For example, the other comparison would be like a qPCR test for mycoplasma, right? Still, people prefer, apparently, the traditional way of testing. So I was wondering your take on when something like NGS will come on board and if the regulatory agencies will accept such a test.
Great question.
Does that make sense? I don't know. It's a long-winding question.
I'll start.
David, you can start.
Yeah, it actually is the top question on the Slido as well. It's kind of related. From what I've heard and what I've experienced is, a lot of, again, that apprehension initially. Well, first off, it is getting accepted, right? So let's start there. It is kind of happening. But I think it's going to be a company-to-company basis, right? Some will try to supplement it. Some will try to do it as an orthogonal test. Some will try to do a full replacement. Haven't heard of anyone fully replacing like the 14-day in vitro assay just yet. I mean, maybe someone knows more than me, but that one is really hard to kind of get over the line. Our probably initial strategy, I'll speak for my group at Regeneron, is we're probably going to target the in vivo test because there is a lot of momentum to replace that, especially within the EU. So it's a good place to start. It's a good stepping stone to show that it works. I'll steal what you said earlier, the fact that, I just forgot what I was going to say. I guess I can't steal it.
It happens to me all the time.
Yeah, apologies. But, oh man, it's killing me. So yeah, so I think different labs will try to do different approaches. I know we'll probably, PCR-based molecular tests can still be a good option. They're really fast, but you will see, I think a lot of labs will be motivated to replace the cell culture-based assays because of the 28-day length. A lot of startups, institutions can't support that in-house anyway, so it takes longer than that because just the logistics and kind of the reporting time contracts that you establish. So I think you'll be seeing once, especially other labs start offering this as a service even, people will jump to it. It's got to have a snowballing effect, and I think we're just getting there. Sorry, that was long.
Yeah.
Sebastian.
Okay. Yeah, with regards to orthogonality, it depends. For replacing, these technologies are good enough to replace in vivo tests. So it does not make any sense to have it as a supplement. Yeah, and the authorities or the regulators that were present and voiced their opinions at the conference, they said, look, we are not demanding an awful lot for you to show that this is good enough to replace in vivo testing. The situation is a little bit different with in vitro, and here they want, well, they don't need a direct head-to-head comparison, but what they really want is some valid data showing how sensitive is my NGS, maybe PCR, but no. How sensitive is the test that should replace my in vitro testing? And here they might ask for a couple more data than for replacing in vivo tests. Yeah.
And I remember now what I was going to say is at the conference, right? They had stated it's not an apples to apples comparison. They're not looking for it. The regulators are already stating we know there's limitations, there's strengths and weaknesses of both approaches. You just need to kind of submit this validation package. Yes, you want to be as sensitive as possible. Yes, you need to check all the boxes. But again, there's that momentum moving that direction. So I think especially this time next year, it's just going to keep snowballing. Okay.
Yeah, if I may add,
Yeah.
Just coming to my mind, if you really look at that scientifically, and as much as I know, in vivo tests have never been that great. I mean, there's maybe one model for one virus that has great sensitivity, but all the other animal-based assays, they don't have the same sensitivity as in vitro used to have. And in vitro always used to be better at detecting most adventitious agents. And there was enough scientific evidence to replace in vivo by other testing methods. But with the advent of these fantastic technologies, we can really, really get rid of animal testing for adventitious agents, definitely.
Yeah, that would be amazing. And Gaston, any points to add from your side?
Yeah, I think coming from where we are, we have a bit of different kind of challenges, I believe, because, well, RNA is either fully synthetic or it is made via IVT, so coming from plasmid DNA. And I think it depends on who you talk to, but if we see the USB guideline that came out, I think the last draft 2 months ago or something, NGS is already part of that guideline. So next to Sanger and RT-PCR as identity attributes testing. So I think it's there already, and how many companies are effectively doing it, I can't say. I've got a feeling that it's still a little bit with Sanger, but it's probably just a matter of time till NGS and ONT therefore is well, the new state of the art.
Great, thanks, Gaston. I see Veronica has a question, so I'll...
Is that okay to ask?
Yeah.
So I just want to add a little bit of flavour, and maybe you've got some more numbers, Sebastian, but I think at the recent PDA conference that you're wearing the hat of, the FDA shared some nice statistics showing that, I think it was between 2020 and 2022, from my memory, if that's correct, they have had in excess of 25 submissions for the use of sequencing for adventitious viral agent testing for cell bank qualification, master seed qualification. So that's right at the cusp of just before the regulations kind of said, yes, go ahead, write your risk assessment, justify why you're going to do it. We don't need to see any comparable data for particularly the in vivo methods, which, by the way, for those that aren't so familiar, that I think the publication showing that type of AVA testing and how insensitive it was, that publication has got to be more than 15 years old, right? It's definitely more than 10 years old.
It's old.
So, this industry has known for more than 15 years that this is not the right way to be testing products for safety, in this case, viral safety, yet we are still doing it. And we've been talking for probably more than 15 years about replacement, moving sequencing into this space. We are now doing it. And Nanopore has and I'm speaking on behalf of Nanopore, obviously, Nanopore has a part to play in that process. So I don't know, Sebastian, if you could add any more flavour in terms of if you have any more recent data in terms of submissions, but I can see that if we have 25 that have been submitted, approved prior to the official revision 2, what's that going to look like? How do you think that? What do you think that would look like over the next coming years? Now they are saying, go for it.
I cannot give you the exact numbers. No. Well, we're coming from classically from a really conservative industry, and as you said, we knew we were doing something that was obsolete and ethically wrong. But no one was really, well, most companies, except Regeneron, they are waiting until others do it successfully, right? And so everyone wants to be a fast second. Well, now everyone sees it's been it's being successfully done, and now we can get on it. And according to numbers, we did a little Mentimeter session, or two actually, during the virus conference, and asking like what aspect of ICH Q5A revision have you already implemented? And 30% answered replacing in vivo by net testing methods.
Excellent.
Yeah, and replacing in vitro came at around 20%. And then we had, what do you want to do next of what the revision enables? And top score was, I want to replace in vivo testing. So it's going to be, it will go quick, but
Yeah.
don't ask me to quantify it.
No, that's great.
Yeah.
And so also for this audience's benefit, for those that aren't familiar, over the, I don't know, the last 30, 40 years, there's been about 26, 27, 28 confirmed contamination events, some of which have been identified by sequencing, or certainly the use of sequencing. How do you see, because obviously we're using these compendial methods and we feel safe using them, however they are inherently unsafe. How do you see that safety level improving with the use of sequencing? So for example, do you think there'll ever be a time, let's say in the next 10 years, where sequencing perhaps plays the predominant role in the safety, viral safety of drugs, where we no longer see any of these contamination events happening, or we find them very quickly as part of an emergency response? We suspect that there's something going on in this bioreactor, we're on it, and then these classical contamination events that have happened in history stop entirely in the future.
Yeah. Just very quickly.
Go on quickly. Yeah.
So there has never been any contamination, any confirmed contamination event that wasn't picked up by another assay. And most contamination events were probably missed by animal models, right?
Yep. David.
Again, I think it's also another question. We can answer another question on here. Is, I mean, our goal is to turn our cell culture-based testing lab into a NGS testing lab. So that's our strategy.
Amazing. Gaston.
I don't think I have anything to add here, actually. So.
Well, then let's take the next one on the Slido, because I think that is to everybody, and it's, if I can get it back, because otherwise I have to remember. What do you think will be that one assay test to rule them all? So you've got it quoted in there. David, you take it away, because you've thought about this, I feel like.
Well, yeah, so we're doing two things, right? So if you're talking about the adventitious agent testing, a metagenomic assay. It captures everything. We're going to go by test code, let's say. So we'll start with in vivo, but in reality, it can replace myco, sterility, anything, right? So that would be the ultimate goal. Yeah, you can detect anything in your sample if you have the right sensitivity. In the gene therapy space, right now, it's just you're getting, again, a wealth of information from each sequencing run. We can really address information responses from the regulatory agencies. We can really well characterize these AAV templates. Seems like we learn something new all the time. It's very interesting. So again, kind of like I commented earlier, just be those critical quality attribute testing, where in one test you're getting all that information that you need, even the safety profile, if you really want to add that on as well.
Amazing. And Gaston, what do you think from your side?
Yeah, I think from our perspective, it's also, again, this multi-attribute that I mentioned already, the 5' cap polyatyl, but especially integrity testing. I think this is something that where we're really looking for something new in the field, because simply that those RNA constructs, they get bigger and bigger. And so people are looking into self-amplifying RNAs, which tend to be tens of thousands of bases long. Right. So, and the longer those things get, the harder it is to establish a good integrity measurement. Simply because, well, traditionally LC-UV or capillary gel electrophoresis don't bring that kind of resolution in order to say, now this is really an integer product. And I think this is where ONT can really deliver extra value that is not there yet. by any of the other techniques that I'm aware of. So yeah, this is probably going to be a very big game changer.
Fantastic. Thanks, Gaston. We've got 5 minutes left, so I'll open it up to the floor again, or I will go to Slido if no one's got anything that they want to ask. Okay, so what I'll...
Over there.
Oh, is there? Sorry.
We got one.
Apologies, we've got one. Excellent. Fantastic.
Hi, Pranay from Gritstone Bio. I'm just thinking of, something like ctDNA, which was, an established technology that regulators needed some help with establishing clinical efficacy. There's, working groups like Friends of Cancer and Blood Pack that have brought industry collaborators together to kind of standardize this technology and bring it to regulators. I would love to get some opinion from the group of the utility of a similar collaborative working group for bringing NGS and CMC, and maybe why we haven't seen any of those yet. Thanks.
Anyone want to kick off? David?
Yeah, if I understood what you're asking correctly, I would say we are doing that. We're part of a lot of different consortium run by like the FDA or EMA. So we're involved directly with the regulators. We're involved with our other biotech competitors even, right? We're all striving to share data and try to push the implementation of NGS as a whole as a suitable method. So I know that's going on from our perspective. It's pretty ubiquitous, I think, right now. But yeah, I don't know if you...
Okay, what was the question again? Sorry.
Do you still have the microphone? Do you mind repeating? No.
Yeah, I don't know if I answered what you were asking, so yeah, maybe repeat the
sure.
question.
Yeah, just more information about, some of these industry-wide collaborative working groups to bring this technology and standardize it in a way that, individual companies are not necessarily having to reinvent the wheel every time they do a submission.
Okay, yeah. I mean, the main one we're involved with... Oh, sorry, real quick. The main one we're involved with is the ABTE group. Serge in his talk mentioned it earlier. That's a great one. Just being involved with IABS or the PDA even. We're not really sharing data.
Well, the FTIC is part of the PDA.
Oh, right. So there you go. They exist.
Yeah.
And from
Perhaps
your side,
the best
Gaston?
example.
Yeah.
Sorry, I think the best example is already here. So ONT and Lonza together are working on workflow that will enable individual customers to simply use as much as they can and are willing to, of course, without having going through the whole developmental stages and the validation stages. So in the mRNA field, I'm not sure if I'm not speaking now for the whole group, but for the mRNA field, these kinds of developments are already ongoing and then enabled by ONT and Lonza together.
Thanks. And we've only got a couple of minutes left, so we'll just have... Oh, we've got one more question on the floor, so yeah, we'll take
if
that.
possible.
Please go for it.
Medini, welcome to part 3. A question for Gaston. You mentioned multiple times integrity. And I was wondering, in your strategy, are you thinking of establishing integrity with ONT de novo? Because I wouldn't be able to say today what's the gold standard. Many use fragment analyzer, but it's challenged. Are you thinking of establishing the assay de novo, or to use something that is already established as a gold standard that you can show you're accurate against?
Yeah, the gold standard these days is indeed capillary gel electrophy, so the fragment analyzer that you mentioned, but probably even more so liquid chromatography, UV, right? And there's a whole plethora of columns and papers and that people are using and working on, but so far what I see is that none of that really gives the required resolution simply because, size. What I mentioned earlier, the size is the limiting factor there. That the columns that are out there simply do not bring that kind of resolution to separate 10,000 from 10,300 nucleotides from each other, right? And actually, when we're being very strict then this is the kind of resolution that you're looking for in order to say, okay, now I've got something that's integer. So basically it has a 5' cap, a middle part, and a poly-A tail. And something that is missing one of those two components, right? Because then basically that is not functional. Whatever you bring into a cell will not have any effect, so it will not be translated into a protein. Not only this, but also the stability is impaired because of that. So what we're looking for is something that really gives us a number that says, okay, from the 10 to the x molecules, we've got 10 to the x minus y compounds, molecules that are totally intact, meaning 5' cap, middle part, poly-A tail. So that is something that we're trying to establish. I don't think we're the first ones to do that, but yes, just to come back to your question, we're indeed trying to establish this de novo, and of course, parallel to that, we will always be running LC-UV and capillary gel electrophoresis assays next to that until, of course, hopefully at some point that it's not necessary anymore.
Thanks so much, Gaston. We are really running out of time. I just want to get one word from each of the panelists to finish off. What can Nanopore do to continue to support you and where we need to get to in the next 5 to 10 years? But very much a quick fire, 1-2 words. David.
On the spot, validation help with the equipment.
Okay. Sebastian.
Don't know if Nanopore can do that. One barrier to implementing this is the fear of false positives.
Okay.
And maybe you can look into follow-up strategies, how to deal with a positive hit. I don't know if Nanopore can do that, but
yeah. Thank you. And Gaston.
Oh, it's really tough because we're now kind of, we're working together on it, so I'm also co-responsible now to bring that together. But yeah, I think
collaboration.
Collaboration. Well, that is already really going well, so I don't think that needs further improvement. But yeah, I mean, I think making it a very plug-and-play assay for everybody. That's probably what makes most sense for also a lot of newcomers in the industry.
Yeah, fantastic. Thank you so much. And unfortunately, that is all the time we've got. So please join me in thanking all of our speakers, David, Sebastian, Gaston. Thanks, everybody. And I know we didn't get through all the questions, so please speak to them in the coffee break. Thanks, guys.